Alright, so this will be the Block 5 review cell signaling component of Dr. Romero's lectures. Let me know if you find these helpful, and maybe in the future I'll be able to do more podcasts and keep these reviews going. Alright, so anyways, I'm going to conclude this slide in both videos. So for the final and for the board, for the final, it's going to be mostly questions you've seen before, or similar questions you've seen before. The board has been customized in the past. I don't know if they're going to continue to do that, but if they're continuing to customize the exam, it will be very representative of the material that you have seen in class. So a couple of points. I have heard that the Exam Master website can be helpful for this. I've heard a lot of mixed things about it. Some people liked it, some people absolutely hated it. If it's something you're interested in checking out, go ahead and go for it and see if it's something that you like. As I've said before, the Rapid Review Biochemistry 3rd edition book, there's nothing that's going to show up on the board exam that's not in that green book, and it's also very good for the class as well. And the last point is for the NBME subject exam, there is a PDF that contains kind of a brief layout and some sample questions for all the different subject exams that you'll take, all through med school. So that's a very helpful item to have there. All right, so let's talk a little bit about intracellular signaling pathways. So what's going to happen is you're going to start off with an extracellular signaling molecule that is going to bind to a receptor protein. And that receptor protein is then going to elicit a cascade of activated signaling proteins within the cell, or it could be proteins or inert, more simple molecules, not necessarily proteins, as we'll discuss in a minute. And then these signaling proteins will then go on to activate metabolic enzymes and cytoskeletal proteins, or alter cytoskeletal proteins. And these changes will be more immediate in the time frame of seconds to minutes. And then these signaling proteins will then also change gene expression, and that's going to be on the magnitude of minutes to hours. So on this slide, we can see sort of a simplistic cartoon diagram of how this amplification and signaling cascade works together. So in this example, we have epinephrine, which is our external signal, which then activates 3 adenyl cyclases, which then activates about 10 cyclic AMPs as a second messenger. And in terms of test questions or things that you may see on the board, cyclic AMP is kind of the quintessential second messenger that you'll see come up. But then it takes 2 cyclic AMPs to activate a protein kinase A, and that's an all or none response. If you have 1 cyclic AMP, it'll do nothing. It takes 2 to activate the protein kinase A. But then in this diagram, each protein kinase A can activate 5 of some enzyme, and then that enzyme will then activate 5 product, and that will elicit the biological response. So you can see with one effector molecule, or one external signal at the top, leads to the activation of a lot of product at the base. So you can kind of think of this as a large triangle with epinephrine at the top of the triangle and product generating a very broad base. And here are some types of intracellular signaling, depending on what your science background is. Some of this may be kind of beating a dead horse, but I'm just going to pretend nobody knows this and just go through it. So we have contact-dependent cell signaling. So this is mostly going to be immune system type cells, your T cells and your B cells. You're going to go over this ad nauseum next year, so don't beat yourself up too much learning about this particularly. But then we have paracrine signaling, where we have a signaling cell that then sends signals to target cells, and those have to be within close proximity of that cell. And then we have synaptic, which is unique to the nervous system, where the one cell's in direct, or really close contact with the other cell, and the signaling molecules are dumped into the synaptic cleft. And then we have endocrine, where the signaling cell is a very long distance, relatively speaking, from the target cells, and then the signals get sent through the bloodstream to the target cells. So we can see here that there's an important distinction between endocrine signaling and synaptic signaling. So endocrine signaling, it sends the endocrine cells send their signals through the bloodstream to cells at some distant site to elicit a response. However, with our neurons, the cell bodies of the neurons could also be a very long distance from their target cells. However, even if no matter the distance, they're still going to be within close proximity to each other, and the signals will be dumped within the synaptic clefts. This is kind of a point where they'll ask kind of a vague question about the proximity or the distance of cells from one another, and they're trying to hint at get you to understand the distinction between neurons and endocrine signaling. So as I alluded to before, extracellular signals can either act slowly or rapidly to change the target cell's behavior. So you have your effector molecule can bind here to this receptor, and you're going to get altered protein function right away in the terms of seconds to minutes. But it's also going to start the process of changing the genetic profile of that cell, which is going to be slow in minutes to hours. But ultimately, you're going to change the behavior of that cell just two different pathways. And in terms of the things that they're going to ask on the tests or things that could come up, is just understanding the distinction between the rapid response and the slow response. So we're going to start getting a little bit more specific about the types of specific receptor types for cell signaling. So as you've discussed a little bit with Dr. Hostler, ion channel coupled receptors, you see these ions bind to the receptor on the cell surface, which opens a pore and allows ions to flow through the channel. We've also got G-coupled protein receptors, which I believe this will be the first time you guys have discussed this, and they're a little bit more complicated, so we'll talk about them a little bit in slides later on. Then we have enzyme-coupled receptors, so you have a signaling molecule that binds to the cell surface of the receptor, and then it activates some sort of catalytic response in the intracellular domain. Or you can have a signaling molecule which binds to the receptor, and the receptor acts on another protein, and then that protein becomes activated. So it can be sort of indirect as well. And then we also have intranuclear receptors, or intracellular nuclear receptors rather, sorry. And these will be steroid receptors and vitamin D receptors. This is an example. So they have to be lipid soluble so they can diffuse across the nuclear membrane and go to their target sites to activate gene expression. So the example Dr. Romero uses for ion-coupled receptors is going to be the nicotinic acetylcholine receptor. So the nicotinic acetylcholine receptor requires two acetylcholines to bind between the alpha and gamma subunits of the receptor. That might be a little bit more specific than what Dr. Romero is going to ask, so I wouldn't worry about that too much necessarily. But the point is that the acetylcholine will bind here, and then this will allow sodium and potassium to flow through the channel. So when we have an action potential along the nerve, we open these voltage-gated calcium channels, and as you may remember from histo, the vesicles that have to bind to like the synaptobrevin complex, so the vesicles can be released into the synaptic cleft, and then acetylcholine will then bind to the nicotinic acetylcholine receptors, which causes sodium influx into the muscle cell and potassium to go out of the cell. Each thing diffuses down its concentration gradient, and then this causes calcium to be released from the sarcoplasmic reticulum. The next type of cell surface receptor that we're going to kind of go over real quick are going to be the G protein coupled receptors. And there's a lot going on with these guys, but in terms of things that are going to show up on the tests, is that you're going to have this extracellular domain that's going to bind the ligand, and all of these G protein coupled receptors have to have 7 transmembrane helices, and then you have this intracellular domain. Sorry, I think I said this was intracellular. This is extracellular domain, and this is extracellular domain. So ligand's going to bind here, which is going to induce a conformational change here, and then you get like a domino effect where these are going to change conformation, and then this, which induces this to change conformation, and that's going to elicit the signal cascade, depending on whichever G protein coupled receptor that you have. One thing I would like to point out, I didn't do it on the previous slides, is that Dr. Romero's questions will mostly come from the boxes that he has added to the slides. So like here, this red box around this picture, you notice he's got a red box around the 7 transmembrane helices. His questions will be pretty straightforward, and it's mostly like identify the type of receptor based on a physical description. So if you see a question that says, that's essentially the gist of the question is, which type of cell surface receptor has 7 transmembrane alpha helices, it's going to be these guys. And they're very important receptors because you can see here in this box, half of all known drugs act through these G protein coupled receptors or their activated pathways. Over here in this picture here, don't bother trying to learn which residues are interacting with epinephrine. That's a very physical biochemistry thing. Don't waste your time doing that. But it just shows that there's a very specific binding pocket that epinephrine is going to fit into. So following along what I said about how these are pretty important, there's a lot that are linked to diseases. We've got 600 mutations that are known to inactivate G protein coupled receptors through these variety of different ways. But things he's going to ask about, again, are going to be the things that he has in his boxes that he has made. So he'll ask questions about rhodopsin and night blindness and the fact that it is a G protein coupled receptor. So like with night blindness, you have an activating mutation in your G protein coupled receptor. So it's active without the ligand, and it causes night blindness. I don't think I've seen him ask questions from this box here. However, I wouldn't say it's outside the realm of possibilities, but it would probably be fairly low yield for him. So once we have our ligand that binds to the extracellular domain out here, and we get this conformational change and then induce the intracellular conformational change, how do these things work? Well, they work by interacting with these heterotrimeric G proteins. And these consist of an alpha, beta, and a gamma subunit. And you can kind of, for your all's purposes, you can kind of think of the beta gamma subunit as one component, although in reality it's 2 pieces. Know that the alpha and the gamma are tethered to the membrane here, so this would be a hydrophobic tail that's embedded in the cell membrane here. And that the alpha subunit is either going to bind to GDP or GTP, depending on whether or not this is active or not. Just like a real simple thing over here, you can see that the heterotrimeric G protein is inactive when bound to GDP, and it is active when it is bound to GTP. And it depends on the nature of this alpha subunit as to whether or not, or as to what type of cellular response you're going to get. I really like this diagram because I feel like it's a really good one and it's simple enough to show you how these G protein coupled receptors work. So you're going to have a ligand that's going to bind here. You're going to activate the G protein coupled receptor. And this is going to induce a conformational change that's going to favor the binding of this heterotrimeric G protein here. When this heterotrimeric G protein binds to the G protein coupled receptor, this is going to cause GDP to be ejected from the G alpha subunit. And then once this G alpha subunit is no longer binding to GDP, it will free itself from the trimer. And once it frees itself from the trimer, it'll bind GTP. And once it binds to GTP, it will then bind to an active effector, which will then start a signaling cascade, or continue the signaling cascade rather. And then this cycle just repeats itself. The G alpha subunit will eventually you'll lose a phosphate here, and you'll get GDP, and that will cause the G alpha subunit to become inactive and bind to the trimeric G protein again. So you can kind of think as the trimer G protein as being inactive, and then G alpha by itself bound to GTP will be the active form of the G alpha subunit. Here are some examples of some heterotrimeric G protein classes. I believe what Dr. Romero is really going to focus on is going to be the G alpha S and G alpha I. So S for stimulatory, I for inhibitory. So you're going, with stimulatory, you're going to want to increase cyclic AMP, and then G alpha inhibitory, you're inhibiting or decreasing cyclic AMP generation. I don't know if he's mentioned it to you guys or not, but one that'll be important for farm, and I can't remember if he did it for us or not. I feel like he did, but I don't remember for sure. It was the G alpha Q and this phospholipase C IP3 DAG pathway, which we'll talk about that specific signaling cascade in a little bit. He'll like to ask questions about some receptor examples for each of these. So knowing that the G alpha S, that beta androgenic, such as adrenergic, such as epinephrine, glucagon, serotonin, and vasopressin receptors are G alpha S, and that alpha 2 adrenergic receptors and muscarinic acetylcholine receptors are inhibitory, and that if he told you to do this, Q would be like alpha 1. So you can see here down in this picture down in the bottom again, we have G alpha S and G alpha I. So if we have GTP here and G alpha S by itself and it binds to adenyl cyclase, we're going to activate the cyclic AMP pathway. But if we have a response here, such as with prostaglandin 1 and adenosine, which is inhibitory, and it's going to activate G alpha I, so this would be bound to GTP, and then it'll bind to adenyl cyclase. This will decrease cyclic AMP generation. And over here for stimulatory hormone examples, you have epinephrine, glucagon, and ACTH. And sometimes he'll ask questions about whether or not these are inhibitory, and which ones are stimulatory and which ones are inhibitory. So another test question favorite is going to be knowing about cholera toxin and the pertussis toxin, because functionally the toxins work in the exact same way. It just depends on whether or not they're going to act on a G alpha S or a G alpha I. So each of these toxins are going to prevent the G alpha subunit from its GTPase activity to cleave the GTP down to GDP and inactivate it. So in both cases, the G alpha subunit will be constitutively active in the presence of this toxin. With cholera toxin, it is going to prevent the GTPase activity of G alpha S. So you're going to have perpetually high levels of cyclic AMP, and this will cause massive fluid loss from diarrhea. If you want to know more about how terrible this is, I'd suggest you look it up. It's very, very interesting, and there's not really a whole lot you do for it except for replace fluids and electrolytes and just kind of wait for the body to get over it on its own. However, pertussis acts on the G alpha I subunit. So same thing, just it's because now this is acting on the inhibitory G alpha subunit, you're going to have decreased cyclic AMP production. So too much cyclic AMP with cholera toxin, not enough cyclic AMP with pertussis. And with pertussis, more commonly known as whooping cough, most of you have probably heard of that. We all get vaccinated in the US for this. Or most everyone gets vaccinated in the US for this. So most of these infant deaths are going to be in other countries where vaccines aren't regularly administered. But yeah, these are very high yield type test questions just for this course and just going forward. So I may have mentioned earlier that it takes 2 cyclic AMPs to activate a catalytic response from protein kinase A, and that's more of a stereochemistry comment. So it takes 4 total cyclic AMPs, and then you get 2 catalytic domains. that'll be active. But you have to have all 4 of these bind for these 2 to be active. So if you see a test question that's getting along the lines of, how many cyclic AMPs do you have to have binding to the regulatory domains for the catalytic domains to be active, it'll be 4. If you have 3, it's not going to do anything. You have to have 4. And then it'll activate 2 of the catalytic domains, and then these catalytic domains will then phosphorylate some target protein, and then that can either have intracellular effects, and then, of course, it can be dephosphorylated to alter its activity back to what it originally was. He will ask about this. So the function of the catalytic subunit can be either will be to transfer inorganic phosphate from ATP to a serine or threonine in a protein, and this is very important that it will be a serine or threonine. And all these catalytic subunits are a little bit different, and they target a very specific sequence and very specific proteins to activate them. So you just don't get this, widespread activation of all the proteins in your body. They are specific. Then we get biological effects. I harped on this a little bit, I believe, during Dr. Hebert's blog. Phosphorylation doesn't necessarily mean the protein's going to be activated. It just entirely depends on the protein. Phosphorylation can turn a protein off. Phosphorylation can turn a protein off, or it can turn it on. It just depends on the protein and which residues you're phosphorylating, but phosphorylation can induce changes in the way that protein or enzyme functions. Phosphatase, we have kinases that are going to add phosphate, phosphatases that remove them. Some other high yield things that he might ask will be that these phosphorylations can block substrate binding, and they can also block protein-protein interactions. So you can think if you have this, highly electronegative phosphate where there previously wasn't one, that it's not going to that's going to cause some steric hindrance. It's not going to want to bind like it previously did. So this slide is from metabolism. Like I mentioned before, it'd be kind of nice to have this lecture about how these G protein coupled receptors work. Along with that, because I feel like it just kind of makes a little bit more sense with metabolism, what gets activated and what gets phosphorylated when you understand how this all functions. So we have a cartoon here that shows that glucagon and epinephrine act through these G protein coupled receptors, and they're going to then activate this heterotrimeric G protein complex. You're going to get an active G alpha subunit, which is going to activate adenyl cyclase. Adenyl cyclase is then going to generate cyclic AMP. And once you get 4 cyclic AMPs, you're going to bind the 4 sites on the regulatory element of pKa and activate the catalytic domain. You'll have 4 of these, or 2 of these rather, sorry. And then you'll start phosphorylating things. So you can see here from the metabolism block, this is what phosphorylates the glycogen phosphorylase kinase A, so active. And then that phosphorylase kinase then activates glycogen phosphorylase A, and then glycogen is degraded. So this is the signaling cascade specifically for glycogen. Other high yield things, I just kind of want to walk through that a little bit. So high yield things on this slide is that these G-coupled protein receptors here will act through the cyclic AMP, and then those activate protein kinases. And then everyone's favorite thing, methylxanthines, such as caffeine and theophylline. Definitely familiar with caffeine, may not be familiar with theophylline. These cause increased levels of cyclic AMP because you inhibit this protein here, or this enzyme here, phosphodiesterase. So you don't break down your cyclic AMP, and your cyclic AMP levels just stay high, and then so everything just stays active. So as we discussed before, a lot of these cell surface receptors that start a signaling cascade will have an immediate response, which we kind of already talked about, but then they will also have a nuclear response as well. That's going to be a little bit longer, minutes to hours sort of time frame. So we see here that we're going to Same signaling cascade. You're going to activate adenyl cyclase. Adenyl cyclase is going to generate cyclic AMP. We're going to have these activated pKa catalytic domains. But for the gene regulation, they're going to go into the nucleus, and they will activate the CREB, which is a transcription factor. I wanted to include this slide in here just to remind you that this, came up earlier in the year in the metabolism block, and now you can kind of see how glucagon works. It works by activating adenyl cyclase. You generate cyclic AMP. You get the catalytic domain activated, which starts phosphorylating things. Then the things it phosphorylates things, and then you get your biological response for the fasting state scenarios. So now that we've got all of these different enzymes and effectors all activated, what do we want to do when we want to turn them off? So one thing that he likes to frequently ask about is arrestin. And you can just kind of think of I think Dr. Aber might have given you guys this example. You've got this hyperactive kid with all these phosphorylations and you want to shut him off. So what do? You hug him. In this case, it's called arrestin. So another thing you can do is also internalization. So you have all these protein receptors on the surface, and you have this clathrin-coated membrane here, just like we talked about, and or we all learned in histology. And then it can be internalized, and that'll turn off the receptors. So just like the vesicles can open them to the top to make them express, they can also be re-internalized and turned off that way. Then we have GTP hydrolysis. So that's what we talked about with the G proteins. The G alpha subunits have some GTPase activity, so they're sort of self-regulating. Second messenger inactivation in protein phosphatases. But really, like the biggest things for him is going to be the GTP hydrolysis and arrestin, I believe. And this is just a quick terminology slide. Knowing that an agonist and what an antagonist is. Sometimes this will show up on the test, and it's really just like a simple vocabulary type question. You definitely don't want to miss something like this because it's basically free points. So an agonist is going to mimic the natural ligand. So this can be an inhibitory response or an activating response. It just depends on what the natural ligand does. Just so don't think that an agonist just by default activates something. Or activates a biological response. Because if the natural ligand inhibits a biological response, then the agonist is inhibitory. So just keep that in mind. Mimics the effect of the natural ligand. However, an antagonist blocks the effect of the natural ligand. It'll do the opposite. So if the natural ligand activates, an antagonist will turn off. Or if the natural ligand turns off, an antagonist will turn on. So just make sure you have that sort of squared away. All right, so we talked about G protein coupled receptors that function through the cyclic AMP protein kinase A pathway. Well, there's another category of G protein coupled receptors, and these will be the G alpha Qs here, which I mentioned before. So maybe you do need to know these. I actually forgot that I had this slide in here. But anyways, so it's kind of a somewhat similar sort of thing. So you have your G protein coupled receptor here, which is then going to activate a heterotrimeric G protein. The alpha subunit bound to GTP, in this case, instead of binding to adenyl cyclase, it's going to bind to phospholipase C. And when it binds to phospholipase C, it's going to cleave this molecule here. And this is a favorite test question here. This guy here, before he gets cleaved, is called PIP2 or phosphatidylinositol 4,5-bisphosphate. So when the G alpha subunit is active and it binds to phospholipase C, this gets cleaved. And then you get inositol triphosphate or IP3 and diacylglycerol, which is retained in the membrane. That's another important factor. DAG will be retained. Inositol 1,4,5 phosphate will then go to the endoplasmic reticulum to bind to a calcium channel. And then it'll release calcium, and then that'll elicit a cellular response. And then up here we have diacylglycerol, which is then going to activate protein kinase C. So don't get this confused with protein kinase A. It's going to function very similarly, but it's, alphabet soup. So if you have the IP3 DAG calcium pathway, it's activating protein kinase C. If you have the cyclic AMP adenyl cyclase pathway, you're activating protein kinase A. And that's a way that the questions will kind of come up to. So try not, do what you can to not get confused with all the alphabet soup. So not really a whole lot to say here about calmodulin and calcium, except for that calcium binds to calmodulin. But very similarly to pKa in the regulatory domain, you have to have 4 calciums binding to calmodulin for it to have its activity. So the question will say something about, you only have calcium at 3 sites, or 1 site's mutated and can't bind calcium, or something along those lines. It will be inactive because you have to have all 4 calciums in the calmodulin complex in order for it to elicit its effect on its target enzymes. And again, this is just a diagram showing rhodopsin, which is something that he's very likely to ask about. Again, with like the night blindness, so you have rhodopsin, which is a G protein coupled receptor, which will then activate a signaling cascade, which activates 11-cis retinal, and you end up with all trans retinal bound to opsin, and this is the active form. So 11-cis retinal not active, all trans retinal active, and that all occurs through a G protein coupled receptor, which is rhodopsin. This slide here is becoming increasingly important in medicine in general and research, and it's probably something that's going to show up. I have a strong suspicion it'll show up on a lot of things, maybe including this test. So acetylcholine will bind to this G protein coupled receptor, which activates a phospholipase C pathway. So this is going to be the IP3 DAG, which is going to release calcium, which is going to then activate nitric oxide synthase. And just kind of like a quick review, that uses NADPH, one of the few pathways that uses NADPH. And you're going to get citrulline and nitric oxide. Nitric oxide will then go to smooth muscle cells, which causes the generation of cyclic GMP, and the cyclic GMP will then activate protein kinase G, which causes relaxation of the muscle cell. Typically the questions for this aren't too brutal, just knowing that nitric oxide causes relaxation of muscle cells through activation of cyclic GMP, or is the highest yield component of this slide. And then this slide kind of highlights why it's important. In the past, it was important pharmacologically, mostly because of drugs for like libido and erectile dysfunction, such as Viagra, Cialis, and Levitra. So these are all PDE5 inhibitors, which you can see here. PDE5 degrades cyclic GMP to just 5 or a GMP. So you lose this activation of protein kinase G that and then induces the smooth muscle relaxation. So let me repeat that. So these drugs will inhibit this, which prevents the breakdown of cyclic GMP, which leads this constitutively active, which leads to smooth muscle relaxation. Normally, this will be degraded by PDE5 so that you lose the relaxation. But the reason it's kind of getting a little bit more important is that we're finding uses for it in things like preeclampsia, and it's just not going to be a sexual dysfunction drug. The next class of receptors that'll be on the test will be these receptor tyrosine kinases, and the example they show here in the picture is insulin. So be sure to know this. This will probably show up in some fashion. And so in the extracellular domain, you've got these two alpha subunits, and then the transmembrane component is the beta subunit. And then insulin binds to this extracellular alpha dimer sort of situation up here. And that's kind of the way I've seen the questions asked before. And then you get, when it binds, you get an immediate response or very rapid short-term response through the IRS phosphorylation, RAS activation. And don't confuse this with RAS, that'll be capital RAS, which we'll talk about in the hormone lecture. This is capital RAS, this different signaling pathway. Other similar pathways to the insulin will be the epidermal growth factor, as well as like HER2/neu, which is important for breast cancer. So he could ask about these as well. Like I mentioned, he may ask for specific examples of these receptor types. But yeah, that's the biggest things, is knowing that you've got the two extracellular alpha subunits, transmembrane beta. Insulin binds insulin, epidermal growth factor or HER2/neu will bind to this extracellular domain, induces conformational change. You get these autophosphorylated tyrosines, which then activate through phosphorylation this IRS tyrosine here. Then you get the cascade of signals. The next class of cell surface receptors that'll show up will be the receptor-associated tyrosine kinases, as you can see here. So these are two transmembrane proteins that are generally separated, and each one is associated with a JAK protein. When you have a cytokine that binds to the extracellular domain and it brings these two transmembrane components together, the JAKs will autophosphorylate each other. And then once they become phosphorylated, they will then phosphorylate the tail of the transmembrane component. Once this becomes phosphorylated, that'll attract the binding of these STAT proteins via their SH2 domain. This might be a little esoteric, but definitely knowing that JAK, STAT go together is important, because that's, what these are called. They're called JAK, STAT. It's called JAK, STAT signaling. And then once these bind, they will also be phosphorylated by JAK. They don't autophosphorylate. JAK directly phosphorylates, or JAK will then cause them to be phosphorylated, rather. So this is not a separate protein here, so don't be confused about that. This is just a domain within the STAT protein. So once these get phosphorylated, you'll have this dimerization here. This is not a tetramer. Again, the SH2 is a domain, so an amino acid sequence within the STAT protein. And then this will then go to the nucleus and activate gene expression. Again, be sure to know these examples of this receptor type here, because he likes to ask about these, and that growth hormone and prolactin are specific examples of JAK-STAT proteins. So another signaling pathway, I think I forgot to mention this for the JAK-STAT, but this is similar too, is that these regulate gene expression, and they don't really activate like quick responses like the G-coupled protein receptors do. These pretty much the signaling pathway goes straight to a gene regulatory element. So this is true for these serine threonine kinases, such as the SMAD signaling, as well as just the JAK-STAT receptors. So here we have, for example, TGF-beta that's going to bind. It's going to bring these two transmembrane proteins together. This serine threonine kinase here is then going to phosphorylate this residue here. Once that gets phosphorylated, this serine threonine kinase is going to then phosphorylate this SMAD2 or SMAD3 protein, which will then associate with SMAD4, and then this will then go to the nucleus to regulate gene expression. So just kind of word association with serine threonine kinases and SMAD2, SMAD3, it's a SMAD signaling pathway. And it's also sort of numerical. You start off with SMAD2 or SMAD3, and then it associates with SMAD4, and then it goes to the nucleus. It's not seeing him ask terribly complicated questions about this. Just again, it's probably just gonna be more word association with just the SMADs. This slide shows things that we've already talked about, where we've got nuclear receptors that in the classical context, where you have some sort of nuclear binding or DNA binding element that will then activate transcription or inhibit transcription in the nucleus. But down here is, like we've been talking about these proteins, is we'll have very rapid responses that are going to activate different kinases, and these will eventually lead to activation or inactivation of genes as well. So you can get these sort of direct response here. You see cortisol binding to the glucocorticoid receptor here, which then directly goes into the nucleus and activates gene expression. Or we can have this membrane pathway, which is these cell surface receptors we've been talking about that have this long signaling cascade that will eventually lead to gene expression being changed. But the quick response is going to be fast, it's going to be short-lived, it doesn't rely on transcription, it's just going to be the activation of these kinases, activation of proteins, and then once they get into the nucleus and do their thing, you get the slow, long-lasting response, and it'll be sort of changing the gene expression profile of that cell. So there's a couple of things that he's asked from this slide that have been a little bit tricky. So an example of a DNA binding domain will specifically be a zinc finger. Not all DNA binding domains are zinc fingers, but an example of one is a zinc finger. And what else here? So all nuclear receptors or ligands for nuclear receptors are derived from cortisol, except for thyroid receptor and retinoic acid receptors. And you can see here you have this variable region, DNA binding region, and a ligand binding domain. And he gives some examples here. So here it's thyroxine receptor and the retinoic acid receptor will not bind a cholesterol derivative, but glucocorticoid, progesterone, estrogen receptors will all bind cholesterol derivatives, as you know, estrogen, progesterone, and glucocorticoids all are built upon a cholesterol skeleton.